right, so for part one of this essay question, we're going to start off with homeostasis. So, homeostasis can be defined as the body's ability to regulate internal conditions. So, an example of homeostasis within the body is body temperature. So, when thinking about the pathway that happens in the body in order to regulate things like body temperature, we start with what's called the control center, which in this case will be the brain. So when there is a stimulus that will go towards the receptor, up to the control center, down to the effector, which will lead to a response, which also provides feedback. So this side of the loop is the afferent pathway, and this side of the loop is the efferent pathway. So with this loop in mind, I guess I'll talk a little bit more about homeostasis using a graph. So thinking about these two different types of feedback, we'll start with negative feedback. So taking the example of body temperature, we will start here and say that this, which is the set point of 98 degrees, is kind of like our main temperature, what we know to be the most common or what is in the middle. What I just drew in the dotted lines is the normal range. So we know that body temperature will fluctuate as different things happen, and so within this normal range, the body temperature will go up and down like this. So what happens is, as the body temperature rises right here, the body will do things like sweat in order to decrease the body temperature. And as the body temperature comes down this way, the body will cause the muscles to shiver in order to increase the body temperature once again. So this loop of the body adjusting its temperature by doing certain things in order to keep the temperature within the normal range is what's called negative feedback. So we want the body will do things in order to keep this temperature as close to the set point as possible by making small adjustments in order to turn it back towards the set point, staying within the normal range. So in contrast of that, I will make another graph here that describes positive feedback. For this example, we can use the oxytocin levels in the body. So here we have another normal range. And for this example, I will talk a little bit about childbirth. So we know that oxytocin levels in the body will fluctuate like this, but when a woman goes into labor, there is more oxytocin produced, which will continue the muscle contractions in order to push the baby out while the mother is giving birth. So this action right here of the levels going continually further away from this set point right here is what's known as positive feedback. Okay, so for part two of this question, we are going to be talking about the types of protein-mediated transport across the membrane. So what I did is I started a little flow chart here that's going to help us kind of explain the different types of transport. So, does it require anything besides kinetic energy? If the answer is no, we have to ask another question: does it need help? If if the answer is no to this question, then we know that we are working with what's called simple diffusion. And this name makes sense because it is the most simple type of diffusion, where this talks about how the ions will go from high concentration to low concentration. Some examples of ions that will undergo simple diffusion are CO2 and oxygen. The rate of this diffusion is dependent on the concentration gradient within the cell. So these happen with small non-charged particles. To kind of give a little bit of a visual here. Here we have a cell here, and we'll say that there's a high concentration of molecules or ions on this side, and this membrane right here will be permeable to these ions, and so they will simply diffuse to the area of low concentration right here. So that is simple diffusion. So if the answer to this question, does it need help, is yes, then we are dealing with what's called facilitated diffusion. So this type of diffusion will also go from high concentration to low concentration, similarly to simple diffusion. The rate is also gradient dependent. And when thinking about facilitated diffusion, that helps us to know that there are different things that are required in order to carry out this diffusion. And so we have different types of channels. First, we have pores, which are also things like leak channels. We have channels that are gated, which include voltage gated, ligand gated, and mechanical gated channels. And so when talking about the specificity of these channels, if we draw a cell here, say we have a channel here that is specific to, or we'll have this channel that is blocked off. It's not open. There needs to be something that happens in order to open this channel. And so this channel could be specific to, I don't know, anything like calcium. So this calcium or another molecule would need to bind to this channel, this opening right here, in order to cause for this channel to open to allow molecules or ions to go in. So each one of these types of channels, these gated channels, require specific molecules or ions in order to open the channels, in order to allow for movement in and out of the cell. And just going along with that specificity, there's also a competition for these specific channels. So when there are multiple ions or molecules that are trying to enter the cell or exit the cell, there's only a certain number of channels and only a certain number of molecules and ions that can bind to those channels in order to allow for flow to happen. So when going back up to this question here, does it require anything besides kinetic energy? If the answer is yes, then we have two different types of active transport. So this we will talk about primary active transport and secondary active transport. So with primary active transport, the main user of this type of transport that we have talked about so far is the sodium potassium ATPase pump. And the reason that this answers yes to, does it require anything besides kinetic energy? There are 3 requirements for this pump to pump sodium and potassium in and out of the cell. And those 3 are obviously sodium, potassium, and this also uses ATP. So drawing a cell here, we can show the pump here, and what happens is when ATP binds to the cell, binds to this pump, we will have the movement of 3 sodium ions out of the cell and 2 potassium ions into the cell. So this is called active transport because it also requires the use of ATP. In terms of secondary active transport, this is secondary because there is an indirect use of ATP. So what happens is this primary active transport will drive secondary active transport because based on the gradient that is created with this pump here, that will determine how the secondary active transport happens as well. So drawing a cell here, we will show that this active transport is going to want to push sodium into the cell against its concentration gradient. And there are 2 different types of this transport here. And so when this sodium goes against this concentration gradient, it has the option of pushing molecules or ions into the cell. We'll say glucose here. And because both of these ions are moving in the same direction, this is called symport. And with this other example here, we can have another molecule or ion exiting the cell, like calcium, and this is called antiport. And both of these actions happening together are called co-transport. And so these are the different examples of the protein-mediated transport that we have discussed in class and how each one of them works based on its requirements of anything besides kinetic energy. We have simple diffusion, facilitated diffusion with different types of channels, primary active transport that uses ATP, and secondary active transport, which uses the concentration gradient provided by the primary active transport.